Hello and good day. In this video, we will learn about contamination. How this contamination looks like, types of contamination, source of contamination, and steps in minimizing contamination. What is contamination in cell culture lab? Contamination is presence of unwanted particles or organisms that may interfere with the optimum growth of desired cultured cells. Contamination can be from microorganisms or chemicals. Let's go through the chemical contamination first. In cell culture, chemical contamination can be from endotoxins, plasticizers, and detergents. These contaminations can affect the growth of the culture cell. Now, what about cross-contamination? Cross-contamination is contamination of microbes which occur unintentionally and transferred from one substance or subject to another with harmful effect. You might be wondering why microorganisms like culture media. The culture media contains nutrients like glucose, ammonia, salts, trace of elements, and micronutrients. These nutrients provide requirement for microorganisms to grow. The presence of serum in culture media also makes it prone to contamination by the microorganisms. This diagram shows different types of microorganisms and they vary in size. If we look at the diameter of the virus, it is the smallest of all microorganisms, whereas eukaryotic cells has the largest diameter of all microorganisms. The shape of bacteria can be observed under microscope, and the shapes can tell us which bacteria might be the contaminant. In this slide, I would like to introduce you to the bacteria that are present in different shapes, such as cocci, bacilli, and spiral. You need to know what are the examples of cocci, bacilli, and spiral-shaped bacteria. How do we identify microorganism contamination? Well, sometimes if they are present in abundance, you can see the contaminants with your own eyes. Example will be fungal contamination. However, to see more clearly and identify the bacterial types, we need to look under microscope under higher magnification. Contamination in cell culture can be from bacteria, yeast, mold, protozoas, and virus. Signs of bacterial contamination. These slides show two flasks with different color of media. Bacterial contamination usually cause sudden change of pH. Aerobic bacteria will make acidic medium change the color from pink to yellow, whereas anaerobic bacteria will cause the medium to become basic and appear more pink than usual. Another sign of bacterial contamination is when you observe the cells under microscope using higher magnification. The space between the cells will appear uniformly granular and may shimmer from the movement of the bacteria. Using the microscope, we can identify the shape and types of individual bacteria. Let's learn in more detail about bacterial infection. Bacteria are usually smaller in size than the eukaryotic cells. Bacteria can appear as rod-like, spherical, or spiral shapes. Under microscopic examination, they can appear as single, in pairs, clusters, or in chains. Some bacteria show active movement, but some are not. How to differentiate types of organism? You need to find and try to focus at one organism and observe closely. For non-moving bacteria, sometimes you need to observe for another several days to see whether there is increase in colony to confirm their presence. Mycoplasma contamination. Contamination in cell culture can also be due to mycoplasma, and these can cause a stress to the investigator as it tends to be tricky. Mycoplasma do not have cell walls, therefore it is unaffected by the antibiotics. The size of mycoplasma is about 0.15 to 0.3 millimeter. Due to its small size, it is easily escaped from the filters with 0.22 millimeter pores. Mycoplasma contamination is not obviously suspected by the naked eyes as they will not change the media color. Mycoplasma can alter cellular metabolisms, slowing the cell growth, prevent cell attachment, and they can influence the results of your study. E. coli contamination is another common bacterial contamination occurs in cell culture lab. A few information about E. coli that you need to know. E. coli is a gram-negative rod-shaped bacteria. The size is about 1 to 3 times 0.4 to 0.7 micrometer in size. It is normally arranged singly or in pairs. It is motile due to the presence of flagella, and some strains are not motile. They are non-sporing organisms. They have a thin cell wall with only 1 to 2 layers of peptidoglycans. They are facultative anaerobes, and they can grow at a wide range temperature from 15 up to 45 degrees Celsius. Some bacterial contamination can also be due to cocci bacteria as a source of bacterial contamination. Have a look at the picture. I hope you could appreciate the presence of small cocci in between the cells. Cell culture contamination can be due to yeast contamination. Yeasts are unicellular eukaryotic microorganisms. They vary in size between 5 to 10 micrometer and up to 40 micrometer. When you look under microscope, yeast appear as ovoid or spherical shape. You can recognize the presence of yeast in your cell culture when looking at the color of your media. Your media can turn turbid, especially in advanced stage. The pH of media may also change, and under microscopic examination, yeast can appear as a single cells or in change form or branches. Next, I would like to discuss on mold contamination. Molds are eukaryotic microorganisms. Molds that grow as multicellular filaments are called hyphae. When contamination occurs, the pH culture media increases and becomes turbid. The mycelia appears as thin wisp-like filaments or clumps of spores. The spores become activated when they are in suitable growth conditions. Mold can appear as whitish or black in culture. They can also look like fizzy patches as shown in the picture. Virus is an infectious agent which is smaller than bacteria in size. Virus can take over the cells and use them to replicate. One thing that you need to monitor is to observe your cells for any morphological changes and also whether there is any cytopathic changes that occur in the cultured cells. How do we identify the presence of virus? Well, the first method is by using electron microscope. Secondly, we can use immunostaining with antibodies. Thirdly, ELISA assays. And lastly, we can use PCR with appropriate viral primers to identify the presence of virus in the culture cells. What can be the source of contamination in cell culture study? Water bath can be the source of contamination if the water is not changed regularly. Contact with non-sterile surface can also be the source of contamination as well. Spillage on materials and on the work surface in the biosafety cabinet can be the source of contamination. And besides that, splashback from pipetting or pouring cell suspensions and reagents can be the source of contamination. Microscopic aerosols and possible infestation by insect, which is rare, but it is possible to occur in cell culture, can be the source of contamination in cell culture work. The last topic that I would like to share with you is how do we minimize the possibility of contamination in cell culture work. There are few steps that can be done to minimize the contamination issues in cell culture lab work. First, apply strict sterile procedure. Wear glove, clean lab coats. Clean the BSC with 70% alcohol before use. Apply UV light sterilization on BSC if available. Use your BAC hood correctly. Clean the incubator and water tray regularly. Clean water bath regularly. Minimize exposure of cells to non-sterile environment. And use sterile labware. Other tips: use filter tips and change regularly. Check your cells often. Bleach your contaminated samples. Spray everything with alcohol. And minimize exposure of your cells or your flask to non-sterile environment. Okay students, I hope by watching this video, you have gained the information related to contamination issues in cell culture work. So I wish you all the best with your experiment.